Hi, everybody. Thanks to OAS for giving us the opportunity to present here. So Skye is focused on unlocking the pharmaceutical potential of the endocannabinoid system and cannabinoids in general. We are a publicly listed company, so I do want to make sure that everybody's aware I may be making some forward-looking statements. Our main focus, and which I'll be sharing with you today, is developing a CB1 drug as a new therapeutic class to treat glaucoma. We know that the CB1 receptor and the endocannabinoid system in general is highly expressed in the eye. CB1 receptors and CB2 receptors are found in the trabecular meshwork as well as the iris ciliary body and have been shown to be impactful in aqueous outflow as well as aqueous production. As we also know, one of the main molecules that activate the CB1 receptors happens to be THC, which is a component of cannabis. And cannabis, as I'm sure a lot of glaucoma clinicians that may be here today, has been shown to reduce intraocular pressure. And I'm sure lots of clinicians have heard from a number of patients, how can I use cannabis to manage my glaucoma? And ultimately, it has been shown either smoking cannabis, orally ingesting cannabis can reduce intraocular pressure by as much as 20 to 30%. But we all know there's challenges here. You have systemic challenges, psychotropic effects, increases in heart rate, decreases in blood pressure, and ultimately the IOP isn't managed for very long, as we know. And in the early days, they actually did try to develop eye drops with THC, which just turned out to be not very effective. And what we know the challenge here is really, again, the safety effects, but also the fact that THC is actually quite a lipophilic molecule, so delivering it directly topically in the eye is very difficult. And that's where Skye comes in with our technology. What we've done is we've taken a synthetic THC and modified it by adding an amino acid, in this case valine, and a hemisuccinate to make a prodrug. This increases the solubility of the drug, making it more formulatable. It also increases the bioavailability of the drug in the eye. And what we've seen in our preclinical models is that when delivered as a topical agent, we can increase the concentration of THC in the eye in the rabbit models, while also being able to reduce intraocular pressure. Importantly, we've also developed a nanoemulsion formulation that also improves the bioavailability of the molecule into the eye. So we've taken this to the phase 1 clinical trials. We conducted a phase 1 study in Australia where we treated healthy volunteers. The focus of the study really was to evaluate safety, because the question everybody is going to ask is, well, if you drop this in the eye as a prodrug, obviously the active agent is still THC. Are you going to have systemic side effects? Are you going to make these patients sick? Are they going to have hallucinations? Are they going to have psychotropic effects? Are they going to get red eyes like people get when they smoke cannabis? So that's what we really evaluated. We did a pretty conventional SAD-MAD study where we dosed 6 treatment cohorts with increasing concentrations at 0.5, 1%, and 2%, and then a MAD population where we dosed with the same increasing concentrations, but we dosed patients twice a day for 5 days. And so what we saw from a pharmacokinetic profile is that we actually didn't see very much THC in the blood at all. We saw some prodrug, but generally the drug did not convert when it entered into the plasma, generally because the enzyme that converts the drug, the carboxylesterases, are not freely floating in the plasma. They're tissue-bound and found in the eye. And so the drug, we did not see any, again, systemic exposure to THC or its more active metabolite, 11-hydroxy-THC. And this coincides with seeing very little systemic side effects. We didn't see any cases of psychotropic effects. We didn't see any increases in heart rate or tachycardia, and we didn't see any decreases in blood pressure. So this was very encouraging for us from a safety profile. Now, the question of hyperemia, we saw very mild cases of hyperemia. We saw a low incidence of hyperemia in this population, in particular the MAD population that received this drug for twice a day for 5 days. We saw a low incidence of below 10%. In fact, it was about 8% of hyperemia, and this compares very favorably to the current drugs. And obviously, this shows that when you drop THC onto the surface of the eye, you don't get those bloodshot eyes that may be characteristic of smoking cannabis. From an ocular comfort perspective, there was some discomfort that we saw, and that comfort was dose-dependent. As we increased dose, we saw more pain, discomfort, stinging, burning in the eye. But what we saw was that the discomfort was transient. So there was discomfort immediately upon installation, but that discomfort was basically gone within 10 to 15 minutes of initial installation. And the question of IOP always comes up. This was a healthy volunteer population. IOP in this population was generally quite low in the group that we dosed. They ranged from around 12 to 17, 18 millimeters of mercury. So we didn't see in general a reduction compared to placebo. But what we did do was a subgroup analysis of this population where we determined kind of a higher IOP baseline. We determined 17 millimeters of mercury in this case as a high baseline IOP for a healthy volunteer population. And what we saw in this group was that we did see a relatively significant reduction in IOP, ranging between 14 and 31% in this group. So although, again, this wasn't the purpose of the this clinical study, we saw this as very encouraging results. That topical drop of this prodrug, SBI-100, has the potential impact of reducing intraocular pressure. So in summary, it appears this drug is safe. We did not see any systemic side effects. The discomfort was transient, and there was a lack of hyperemia in this population. And importantly, it appears that at least in the subgroup population, we were seeing some reductions in intraocular pressure. So next steps for us, I'm glad to report that we just initiated a phase 2 proof of concept study. This is a relatively small study. Again, this is just to answer the next question: does this actually reduce intraocular pressure? So we've started this study, 3 sites, 2 here in California, 1 in Pennsylvania, sorry. Short dosing period, 14 days. We'll be measuring diurnal IOP changes from baseline to day 7 and day 14. We're expecting to report interim data in the early part of next year and complete the study before the first half of next year. And that's it. I appreciate everybody listening. Again, thank you for the opportunity.